Okay. Oh no, this is right over where I need it to be. Oh, it didn't help. Oh, hold on 1 second. The Zoom is right over where I need to click full screen for the PowerPoint.
Oh no. Wait, what about the bottom?
Is it full screen for you guys?
Oh yeah, now it is.
Okay. Whew. All right. Let me just get that out of the way. Okay, so hello everyone. Today I'm going to be doing my presentation on my mushrooms project for my mushrooms class. And today we're going to see if maybe mushrooms can help treat sepsis. So first I wanted to talk about what sepsis is. We've all heard about sepsis in movies when someone gets seriously injured and someone screams, "We got to get this guy to the hospital," or before they go into septic shock. So this septic shock, or also called sepsis when it's less severe, is basically your body's immune system getting dysregulated and overreacting to an infection. And symptoms can include a fever, shivering, clammy skin, extreme pain, and disorientation. And it all begins when bacterial infection occurs and some of that bacteria gets into the bloodstream. And so as the bacteria moves throughout our system, it sheds various pathogen-associated molecular patterns, or PAMPs, such as lipopolysaccharide, or LPS, which is found on the outer membrane of gram-negative bacteria, such as E. coli. And this LPS can trigger the innate immune system through the pathogen recognition receptor, such as TLR4. And so once TLR4 is activated, the cascade begins by activating these downstream components and ends here where I-kappa B gets phosphorylated, which causes it to degrade, allowing NF-kappa B to enter the nucleus and initiate transcription of various inflammatory genes, such as pro-inflammatory cytokines and interferons. And so these pro-inflammatory cytokines are like little messenger molecules that alert the neighboring cells of an infection. And so cytokines are responsible for raising your temperature and causing infected tissues to turn red and swell in response to an infection, meaning that the symptoms associated with infection are actually caused by your body's immune response. For example, the cytokine IL-6 induces fat cells to metabolize, which makes heat and raises the temperature in infected tissue, while IL-1 beta and TNF alpha cause blood vessels to become more leaky, allowing effector cells like macrophages to enter infected tissue and help fight infection by phagocytosing bacteria. And so these are all fine and dandy immune responses, especially when the bacteria is still extracellular. But things can get really bad when the bacteria or other PAMPs get inside the cell. And so intracellular LPS can activate cytosolic caspases, such as murine caspase 11 and human caspase 4 and 5. And these caspases are like little scissors that cleave gasdermin D, which causes it to oligomerize and form pores on the plasma membrane. And these pores are large enough for molecules like cytokines to get through, causing a cytokine storm, which I'm sure most people are familiar with now due to COVID. And these pores also drive pyroptosis, which is a form of programmed cell death due to inflammation. And so if enough cells die, then that can lead to tissue damage. And if the tissue damage is severe enough, then that could inhibit the infected organ from functioning properly, leading to organ failure. And if an important enough organ fails, such as your heart or your liver, then you die. And so this is why sepsis has such a high mortality rate, with 25% mortality rate for septic patients and a 40 to 50% mortality rate if they go into septic shock. And so sepsis is the leading cause of death in intensive care units and is responsible for causing around 1.7 million hospitalizations and 270,000 deaths annually, according to the CDC. And so there is no known cure for sepsis. And since many septic patients arrive at the hospital when organ dysfunction is well underway, there are very few treatments that can help at that point. And so this is why it's so important to study sepsis in order to better understand its pathology. So what makes fungi an interesting candidate? Due to their sessile lifestyle, they really only have one mode of defense, which is chemical defense. And they're often found on nutrient-rich substrates. So they're at constant war with competing bacteria and other pathogens. And so in order to kill off or at least inhibit the growth of their microbial enemies, they often secrete antibacterial peptides and proteins to keep their competition at bay. However, many mushrooms rely on predation by various animals, especially mammals, to disperse their spores. So they have to find that delicate balance between having the ability to attack bacterial enemies without harming their spore dispersing friends. So this leads to them to produce a wide array of chemical compounds that can harm bacteria and other pathogens without harming us and may even be beneficial to us. A good example is penicillin. And so many of these chemical compounds produced by fungi have also been shown to have antioxidant and anti-inflammatory properties, which is what we'll be discussing today as it relates to sepsis. So here are the mushrooms that we will be exploring. We have Agaricus blazei, Antrodia cinnamomea, and Polyozellis multiplex. So the first mushroom we're going to be looking into is Agaricus blazei. It has a number of common names, such as mushroom of the sun, God's mushroom, royal sun agaricus, and Princess matsutake. It can be found in the northeastern US, in Canada, California, Hawaii, the UK, Taiwan, the Philippines, Brazil, and Australia. And in this first study, they chose this mushroom because it has been previously shown to have a number of immunologically relevant bioactive compounds, such as beta-glucans, which can stimulate immune responses as well as increase phagocytosis and lymphocyte proliferation, and phenolic compounds such as gallic acid, ascorbic acid, and pyrogalol, it's fun to say, which have antioxidant capabilities. And so the way that Navigantes Lima et al. Made their treatment was by boiling dried and ground A. blazei and then filtering and freeze drying that solution to make an overall stock solution that they used in their experiments, which I thought was really interesting because it was a pretty easy way to make the solution that they made compared to the way other studies make their mushroom extracts. And so here's their overall experimental design. And so they, their way of inducing polymicrobial sepsis was through a procedure called a cecal ligation puncture. And a cecal ligation puncture, or CLP, as the name implies, is where they puncture the cecum with a needle and then squeeze out a little bit of what's in there and then sew the mice back up. And so up here you can see what the experimental groups were. And so the sham group was basically just opened up and then sewn shut without puncturing their cecum and was basically used as a control for the surgery. The CLP and saline group was used as a negative control, and the CLP plus CEF was a positive control because CEF is ceftriaxone, which is an antibiotic. And then finally, we have our experimental group, which is the mushroom solution that they used, which they denote as AB for the rest of their study. And so they gave Swiss mice their solution 24 hours before CLP surgery, and then again at the time of surgery, and then monitored them for 12 days for survival. And then they repeated the treatments, but then euthanized the mice after 12 and 24 hours post-CLP to analyze their bacterial load, oxidative stress, leukocyte migration, cytokine levels and phagocytic capacity. And so the first thing that they did was to see if their mushroom solution had any sort of protective effect on sepsis. And so here is the mice's survival rate and body weight over time. And here you can see that all of the mice that were given the mushroom solution survived, while only 40% of the mice treated with the antibiotic solution survived, and all of the saline mice died. The star indicates significance between the CLP groups and the sham group, while the pound symbol or hashtag means significance between the two CLP groups. And so next you can see that the treated groups weight was initially trailing with both the control groups for about the first few days or so. But then by day 4, the mushroom group started improving, and towards the end of the time period, their weights actually started to almost match those of the sham group. And
Hey,
so,
Rachel?
oh yeah.
Did they use just beta glucans as like a
Oh, they
control?
for like the sham group, or which
I'm
control?
just thinking, I'm just thinking, beta glucans from fungus induce trained immunity. And I think trained immunity could be protective in this model. Like if you hit the mouse, if you give the mouse beta glucans, it kind of primes it for sepsis, and it could cause, I guess, better outcomes. And I'm just wondering if their slurry, the bioactive function of the slurry is just the beta glucans in there.
I think they were hoping to get the beta glucans and the phenolic compounds as well for the antioxidant properties. Because yeah, they just took the entire mushroom in this case and ground it up and used it, as opposed to trying to like isolate specifically the beta glucans or any of the other compounds.
Yeah, I just think it would be interesting if they did just the beta glucans. Like, is it just stimulating the immune system through a pattern recognition receptor that like responds to beta glucans, or is it actually this specific mushroom and all of its compounds? That I feel like if it was this specific mushroom and all of its compounds, including its antioxidants, that would be really cool. But if it was just beta glucans, it'd be like, okay, well, I don't know. That's why I'm not sure if they talked about it.
Oh yeah, yeah. No, they for their study they don't isolate anything, but yeah, that would be really interesting to see. Um, yeah, and so after they did those experiments, they wanted to look at metallurgical parameters, and they saw that for the white blood cells, neutrophils, monocytes, and platelet count, that the mice that were treated with the AB extract almost matched perfectly with the sham groups for each of these.
Is this interperitoneal cells, or where are these cells from?
I believe they're IP cells. Yeah, peritoneal lavage. Yeah, or from the blood and peritoneal. So I believe these were from the peritoneal. And so, next they wanted to look at bacterial load, and saw that pre-treatment allowed the mice to have almost complete control over bacteremia after 24 hours post-infection in both the blood and the peritoneal cavity of these mice.
Wait, can you go
And
back one? Sorry.
yeah,
So they have less neutrophils, but more bacterial killing? Okay. That just seems counterintuitive to me, but I just wanted to make sure. Am I the only one here? Like, that seems weird.
Which ones have less neutrophils? Are you talking about the sham?
I'm talking about the mushroom treated ones, the AB
Oh.
group. They have less neutrophils and less monocytes and less white blood cells in general, but they have less CFU, which means I guess the mushrooms are directly killing the bacteria because they're not getting any cellular help,
Huh.
which is
Well,
weird.
that's really interesting. Yeah. Okay. So yeah, they saw the reduction in bacteria in both the blood and peritoneal cavities. So they wanted to look at cytokine expression next. And so they saw a significant decrease in IL-1 beta and TNF alpha in the serum of treated mice, but not in the peritoneal cavity, which I thought was pretty interesting. They just glossed over that in the paper. They didn't really go over it too much. Yeah, I don't really know why that is, to be honest. What do you guys do? But yeah. So next they wanted to look at the total antioxidant capacity of the serum, the peritoneal fluid, heart, and liver of these mice, since the mushrooms are thought to possess antioxidant capabilities. And so they analyzed antioxidants using a Trolox equivalent antioxidant capacity assay. And they saw that pre-treatment with the agaricus solution significantly raised antioxidant capacity in pretty much all cases. And in some of these, even higher than the sham group, such as 24 hours post-infection for the serum and the peritoneal fluid and the liver, all at 24 hours. So I thought that was pretty interesting.
I just had a quick question on these slides, and I guess the next ones too. Is the hashtag in this case that indicate higher significance than the asterisk?
So the hashtag is significance between the two CLP groups, and the star is significance between the sham group and one of the CLP groups.
Perfect. Thank you.
Yeah.
Hey, Rachel. What is
Yeah.
the TEAC? Like, we're seeing that on all the y-axes. What does that mean that we're actually looking at?
So that's just what this assay is called. I don't know exactly how they quantify the antioxidant capacity. I don't really know too much about that assay, but the TEAC is basically just kind of like the number that's associated with this assay.
Okay, I see. Thanks.
Oh, Trolox. Yeah, I used to, during my undergrad, I did antioxidant assays, and we used that.
Oh, nice.
So what does it measure, Jesse?
It's a vitamin E analog, so I can't remember the specifics, but yeah, it's used to measure antioxidant capacity. The colorimetric reagent.
Maybe it's like how many oxidants it quenches or something.
Yeah, it's a quenching reaction.
Okay.
Okay. Cool. So next, they wanted to look at whether or not the agaricus treatment could reduce oxidative stress markers like reactive oxygen species, DNA damage, and MDA. And so the top is peritoneal macrophages taken from mice and analyzed for the production of reactive oxygen species. And they saw decrease slightly below sham levels in ROS production 24 hours post CLP. And then at the bottom, they did an in vitro analysis of ROS production in mice macrophages again, and incubated the macrophages with TBHP, which induces ROS production, and as well as their mushroom treatment. And they saw a significant inhibition of ROS production when these cells were treated with the AB extract. And so next they wanted to look at DNA damage in PBMCs of healthy donors. And they did this by doing a COMET assay. And so here's the control, which is just regular media. And then the TBHP treated PBMCs, you can see looks like a comet. And that's because damaged DNA due to oxidative stress can cause the damaged DNA to migrate out of the nucleus, which forms this tail. And so you can see that the TBHP group does definitely cause quite a bit of DNA damage, while treating the cells with the AB extract was able to inhibit this TBHP-induced DNA damage. And so finally, they looked at MDA levels in the serum of septic mice, since it's a common lipid marker of oxidative stress, and found that pretreatment with the AB extract significantly decreased MDA levels in the serum. And they also repeated this in the peritoneal cavity, heart, and liver, but all the graphs looked the same, so I didn't want to include all of them, but they did do it. And so moving on to our next specimen, wanted to talk about Entrodia cinnamomea. And so it doesn't really have any common names, but it's a polypore fungus that can only be found in Taiwan. And it parasitizes the inner heartwood wall of the evergreen. Oh, 1 second. I need to plug my computer in real quick, I guess. Hold on, I'm going to resume. Okay, hopefully everything's okay still. So this mushroom so it grows on the heartwood wall of the evergreen tree Cinnamomum kenihire, which, as you can guess, is sometimes harvested for cinnamon. And since this fungus likes to grow on the heartwood of the tree, damage done to this fungus is often referred to as heart rot, which I thought was really adorable. And it's commonly used in traditional medicine as a remedy for cancer, hypertension, and hangovers. And we're going to be talking about 2 specific compounds produced by this fungus known as 4-acetylenteroquinol B and xanthoic acid A. And so first we're going to look at a paper by Chang et al. From 2018 that looks at 4-AQB, which has been chosen by these researchers because it has been previously shown to have anti-cancer properties, as well as inhibit nitric oxide production in LPS-stimulated murine macrophages. And so they wanted to explore any potential anti-inflammatory properties it may have in response to infection. And so they first pre-treated raw cells or peritoneal macrophages with 4-AQB, or they used dexamethasone as a positive control for 30 minutes before treating with LPS, and then looked at TNF alpha and IL-6 concentrations in the cell supernatants. And so here we have TNF alpha on the top and IL-6 on the bottom, with raw macrophages here on the left and peritoneal macrophages on the right. And then on the x-axis, we have a couple controls, which they didn't explicitly explain, but I'm assuming that the S weren't stimulated with anything at all. The CTL control was just stimulated with LPS and nothing else. The vehicle, they did mention, that's just DMSO. And then there are the groups that were pre-treated with the 4-AQB, as well as the positive control DEX. And they saw a somewhat significant reduction in TNF alpha and IL-6 in the raw cells, especially at higher doses. But they saw a significant, like a very significant reduction in cytokines in the peritoneal macrophages. And so they knew that 4-AQB was inhibiting production of pro-inflammatory cytokines. So next they wanted to find out why. So they looked at other key players in the TLR4 pathway, since this is one of the main pathways stimulated by extracellular LPS. And so they looked at the production of nitric oxide synthase, ERK, P38, J&K, I-kappa B, as well as NF-kappa B, and STAT1. And so for how they treated the cells for the NOS, they took raw cells and pre-treated them for 30 minutes with the 4-AQB, and then added LPS and incubated the cells for 6 hours. And then for the MAP kinases, NF-kappa B, I-kappa B, and STAT1, they incubated the raw cells for 30 minutes with the treatment, and then added LPS for another 30 minutes. And for the ERK, they used peritoneal macrophages, and they stimulated them with the 4-AQB for 30 minutes and then LPS for 30 minutes. And they saw a significant reduction in the expression of the nitric oxide synthase, as well as the MAP kinase products like phosphorylated P38, phosphorylated J&K, NF-kappa B, I-kappa B, as well as STAT1. And they also saw a significant reduction in the phosphorylated ERK in the peritoneal macrophages. And so finally, they wanted to see if this had any protective effects in vivo. So they looked at survival and tissue damage using the CLP-induced sepsis model. And so they took ICR mice and IP injected them with either 0.3 or 3 mg per kg of the 4-AQB extract for 30 minutes before CLP surgery, and then monitored their survival for 10 days. And they saw a significant increase in survival with the mice receiving the higher dose having a 100% survival rate, and with the lower dose not trailing too far behind, compared with only about 30% of the control mice surviving. And so then for the tissue damage, they pre-treated the mice again with an IP injection of 4-AQB for 30 minutes prior to the CLP, and looked at histological damage 24 hours post-operation. And they found that the treated mice displayed less glomerular hypercellularity in their kidneys, which is characterized by an increase in the number of nuclei in response to infection, like you can see here in the CLP kidney, and how that goes away with treatment. And they also saw less leukocytes infiltrate the liver and lungs of the treated mice, which you can see here with the arrows for the liver, as well as the wall thickness for the lungs. You can see a decrease in the alveolar wall thickness. And so next I'm going to briefly go over another interesting bioactive compound found in A. cinnamomea, known as xanuic acid A, or ZA, which is shown here. And it's been previously shown to display anti-inflammatory activity by blocking NO production and COX-2 expression in microglia cells. And so in this paper, Chen et al. In 2014 set out to see how it was modulating inflammatory responses. And they did a lot of the same experiments as the previous study I talked about and found similar results, which I'm not going to show all of them, but they did find a dose-dependent reduction of the expression of NOS, phosphorylated I-kappa B, NF-kappa B, ERK, J&K, as well as P38. And since these are all involved in the TLR4 pathway, they wanted to see if perhaps the disruption occurred up there. So they looked at MD2, which forms a complex with TLR4 and activates that pathway when it binds to LPS. And so they used a program called HotLig and XScore, which are knowledge-based scoring programs that were used to predict the molecular interactions as well as the binding affinities between ZA and MD2. And so here you can see a clipped surface model of MD2 along with the ribbon model. And here is the LPS binding pocket. And so this program was able to show that ZA was able to fit in that binding pocket. And they're also able to see which part of the LPS molecule ZA was imitating in order to bind to MD2. They were also able to see which hydrophobic amino acids in MD2 were involved in binding to ZA, as well as which ZA atoms were involved in hydrogen bonds with these amino acids, which are the atoms in red, which I thought was really interesting. And so for their X score for the binding affinity with MD2, it was higher for ZA than it was for LPS, which indicates that ZA could potentially outcompete LPS for MD2 binding. And so they wanted to look at cytokine production. So they pre-treated raw cells with ZA for an hour and then treated them with LPS for 4 hours and collected the supernatants and looked at TNF alpha and IL-6 production. And so here on the x-axis, we have our controls where they either received nothing or only ZA or only LPS. And the two different colored bars indicate high and a low dose of LPS. And you can see that ZA was able to decrease the cytokine production in these raw cells in a dose-dependent manner. So next they wanted to look at cytokine levels in vivo. So they used these C3H hen and hedge mice, which I've never heard of before. And they pre-treated them with an IP injection of ZA 30 minutes prior to an IP injection of LPS. And they saw similar results in vivo as they did in vitro with a dose-dependent decrease in cytokine production for both TNF alpha and IL-6. And so finally, they wanted to look at bacterial load. So in vivo, so they pre-treated BL6 mice with for 30 minutes with the ZA before orally administering this PCMV loop transformed bacteria. And so these bacteria are given a plasmid that causes them to fluoresce when it is in the presence of D-luciferin. And so 48 hours post-infection, they IP injected the mice with the luciferin and did a bioluminescence imaging on them to view their bacterial load. And they saw almost complete clearance of the bacteria in the ZA-treated mice with only just a few areas getting illuminated. And so the last mushroom we're going to talk about is Polyozellus multiplex. It is also known as the blue chanterelle, but it is not in the order Cantharellus. So it's a bit of a misnomer, but it's still a really nice edible. And it can be found here in Oregon, as well as Maine, Colorado, New Mexico, Alaska, parts of Canada, as well as China, Japan, and Korea. And previous studies have indicated that this mushroom may display suppressive effects on cancer, especially stomach cancer, as well as Alzheimer's, and it has antioxidant capabilities. And so one of the main bioactive compounds in P multiplex is called polyozellin, or POZ. And here is the molecular structure of it. And so this lab put out a few papers on polyozellin's role in sepsis, but I'm just going to go over one that focused on HMGB1. And so they thought that HMGB1 would be a good target because it can bind to RAGE as well as TLR2 and TLR4 and activate NF-kappa B through that. And they also knew that accumulation of HMGB1 during sepsis has been found to lead to multiple organ collapse and mortality. And in addition, it is a late mediator in sepsis, which makes it a more realistic target for treatment, since you kind of have to know when you're going to get sepsis for the other treatments we've talked about to be fully effective, which you can't really predict. So first, they wanted to look at the effects of polyozellin on HMG or on the expression of HMGB1 in response to LPS treatment. And so they stimulated human umbilical vein endothelial cells, or HUVECs, with LPS first for 16 hours, and then treated them with pause for 6 hours, and looked at HMGB1 concentration in the supernatants. And they found a dose-dependent decrease in the HMGB1 release when the cells were treated with pause. And so then they wanted to see if HMGB1 expression was still decreasing in vivo in response to treatment. And so they did a CLP on BL6 mice and gave them an IV injection of pause 12 hours post-surgery, and looked at serum levels of HMGB1 24 hours post-treatment. And they saw another dose-dependent decrease in HMGB1 when treated with pause. And so then they wanted to see the effect of pause on HMGB1 cell surface receptors. So they activated HUVECs with HMGB1 for 16 hours and then incubated them with pause for 6 hours and looked at TLR2, TLR4, and RAGE expression. And they saw pause was able to reduce the expression of all 3 HMGB1 receptors in a dose-dependent manner. And so then they wanted to see if pause had any effect on barrier integrity. So they treated HUVECs with either LPS for 4 hours or HMGB1 for 16 hours prior to pause treatment for 6 hours. And they found that treatment with pause decreased both LPS and HMGB1 mediated disruption of barrier integrity, which is also seen here, where they stained F-actin with immunofluorescence and treated HUVECs with HMGB1 for 1 hour prior to pause treatment for 6 hours. And you can see here what healthy, normal HUVECs look like next to some highly permeable cells that manifested the formation of paracellular gaps with HMGB1 treatment alone. And you can see here how the increasing levels of the pause treatment were able to significantly decrease the formation of these paracellular gaps. And so they then tested this in vivo, where they IV injected BL6 mice again with HMGB1 16 hours prior to pause treatment, which they gave for 6 hours. And then they IP injected the mice with albumin-bound Evans blue dye to see how much of that dye leaked out. And they saw a significant decrease in the amount of dye leakage in pause-treated mice. And so next they wanted to look at cytokine expression. And so they treated the HUVECs with HMGB1 for 16 hours and then treated the cells with pause for 6 hours. And they saw a dose-dependent decrease in TNF alpha as well as IL-6 release in the supernatants. And they also saw a decrease in P38 and activated P38, ERK1 and 2, as well as NF-kappa B in the nuclear lysates. And so since they saw that pause had an anti-inflammatory effect through the inhibition of the NF-kappa B pathway, they wanted to see the results in vivo. So they injected the mice with polyocellin at both 12 and 50 hours post-CLP and monitored their survival for 5 days, or 5.5 days. They initially tried this experiment with only 1 dose of pause at 12 hours post-CLP, and all the mice died. So they tried giving them another dose 50 hours post-surgery, and it ended up working out a lot better. About half of the mice survived with the higher dose. Only around like 35–40% of the mice survived with the lower dose, but all of the control mice died. So there was some protective effect. And finally, they wanted to look at the lungs of the mice that underwent the treatment 4 days post, or 4 days post-surgery. So they received the 2 doses of the pause treatment at 12 and 50 hours post-surgery. And then 4 days post-surgery, they euthanized the mice and looked at their lungs. And they saw a significant decrease in the leukocyte infiltration with the treatment compared to the CLP mice. And so, in conclusion, all the mushrooms you looked at displayed some sort of anti-inflammatory effects. Ablasii and A. cinnamomea were only shown to be effective as a pretreatment for infection. However, P. multiplex was found to be successful at reducing inflammation or inflammatory markers and protecting mice post-infection with proper dosage. So I was curious, would you guys take any of these mushrooms? Anyone?
Maybe if the situation was dire.
Yeah, that's what I kind of feel too. Like if I was mushroom hunting and I found P. multiplex, the last one we talked about, the blue chanterelle, and I was on a trail and got hurt really bad or something, then I'd probably just start, well, you have to cook it first. So I'd probably try to build a fire and then cook the mushroom and then eat it like crazy to try to not get sepsis.
Is multiplex a known, like, is that one that mushroom hunters usually go for eating on a regular basis?
I think so, yeah. It was described as, what was the word? I forgot what was used exactly, but one of the main mushroom hunters, David Aurora, described the mushroom as not anywhere near as great as regular chanterelles, but it's still pretty good edible. I would eat it. I would definitely eat it.
Why do you
I
have
think
to
also just because. Oh, why do you have to cook them? I forgot the exact reason why, but in my mushrooms class, they're saying that you should just cook every mushroom, because a lot of mushrooms, even like the little white button mushrooms, some people can have an adverse reaction to that when they're eaten raw. I mean, it's usually just like a stomach ache or something like that, not, you know, serious, like you're not gonna get poisoned or anything, but you might just get like a little upset tummy.
So could you eat any mushroom as long as it was cooked?
No, definitely not.
I heard a rumor that
Yeah,
you should just like not eat any mushroom you find an organ that has gills. Is that true?
For you guys, yes, I would definitely say that. It's really hard to identify gilled mushrooms, and there's so many lookalikes. Usually when I go mushroom hunting, I look at the gilled mushrooms, but I'm just like, oh, that's a pretty mushroom. I'm just gonna go along because I don't know. There's a lot of gilled mushrooms that can kill you. It doesn't take a lot to do it.
Yeah, I was thinking, you know, the cause of sepsis is an infection. And so even though I understand that like a massive cytokine storm is bad for you, you'd think if you took like an anti-inflammatory mushroom while you had an infection, that also wouldn't be very good. I mean, this is like kind of like what people run up against with treating sepsis, because they're like, oh, we should treat the inflammation, and then they treat it and then they get super sick and die of pneumonia because they're like treating they are treating sepsis, but the person's usually infected with something at the same time. So I wouldn't take it, but maybe I would take it if I had a headache or a fever.
Yeah.
So it's interesting to me that they have these anti-inflammatory properties, but then I wasn't even aware that beta-glucan is something that comes from a mushroom, but beta-glucan induces inflammation, doesn't it? So do only some of them have beta-glucans?
Yeah, I don't know if that's across the board. Yeah, I don't think that's inherent to all fungi, but I mean, I'd be interested to see. I mean, even just which fungi have beta glucans and which ones don't would be really interesting, just to see if there's some sort of pattern there.
Yeah, maybe it was just that first mushroom that you talked about with that first study.
Yeah, that's the only one that mentioned beta glucans. The other studies, they didn't really go into it. I mean, they were busy talking about the compound that they were trying to study, so I guess that kind of makes sense. But I don't know. It'd be interesting to look into.
Maybe we just need to find the perfect mixture of mushrooms that have anti-inflammatory and pro-inflammatory properties, and then give that cocktail to mice.
Oh, that would be super interesting. Just take all three of these, grind them up, and throw them at all the septic patients. Like, here you guys go.
Or maybe these would be good for like an autoimmune disease, where you get like unwanted inflammation.
Oh yeah. Either way, this was all very cool, and I had no idea that people were looking into mushroom components to treat sepsis. I think that's awesome.
Yeah, this was great.
Yeah, it was fun to do the research for this.
Would you take any of these, Rachel?
I mean, I feel like if I had sepsis, I would probably be doing just anything I could to not die. So, yeah. I mean, I don't think it would really do anything, but it would make me feel better, like up here.
Placebos are real. And you'd be getting
Yeah. Yeah, I mean, you know, there's minus the middle one here. I don't think that one's supposed to taste very good. It's polypores usually tend to not be very tasty. But I mean, these two are supposed to be very, very good tasting. So, I mean, I would just eat them for fun. Sepsis or no.
Thank you,
Thanks,
Rachel.
Rachel.
Yes,
thank you.
Yeah.